Okay, so as I mentioned last time, Thursday's lecture has to be canceled due to a scheduling snafu with a PI meeting that I would much rather be here for rather than in frigid DC. But I'll be back Thursday night, so then we'll move on ahead. So instead of Thursday's lecture, what I want you to do is to use that time I've just released a pre-lecture 1D perusal assignment that is available online. An announcement will go out at the close of this class, reminding you of this. And so this is a short chapter that will cover some of the things that we will start to introduce in lecture 1D next week. So it's an opportunity to kind of take what we talk about today, extend it a little bit, and prep for the innards of the genetic algorithm next week. So that and it's this perusal assignment is set up as a concept check. And so now I've changed it so that 4 concept checks will be dropped instead of 3. So if this is something that you don't want to do, then that's fine. You could just let it go to 0 after the due date, and then it'll just get dropped, and you'll be back to the 3 concept checks. But if you instead do want to use this lecture time to do that, then it gives you 1 less concept check to do later in the semester. So that is due Sunday night, along with the Muddiest Point for this upcoming Sunday, which will just be over today's lecture. And the other thing is the concept check 1A, I originally was planning on having to do Sunday. It probably still, the content would fit with Sunday, but it normally comes after the content that I'll cover on Tuesday. So I've shifted it to Wednesday of next week. So the only thing due this weekend is the Muddiest Point and this pre-lecture perusal assignment. How many of you have done assignments in perusal before? Anybody? So this is, I think, about 1 or so. This is a tool that I use in a lot of my reading-based classes. I really like it. It uses an AI-based grading scheme. So basically, you'll open the assignment and it'll just open the chapter. And from the chapter, you'll be able to read through it. And there are a bunch of different ways that you can score 100%. So you can't go over 100%, but the multiplicity of the ways to get 100% is very high. So you get a few points for opening the assignment, a few points every time you open the assignment, a few points for scrolling down through the assignment and reading the chapter. You get significantly more points for leaving annotations on the chapter that other students can read. You get also points for responding to other students, and then you get also points when people respond to you. So if you just do one of these things, you can't get the 100%. But if you say leave a few high quality comments, and then it also scores them. So it has sort of an AI based classifier where it'll have like a high quality comment, a medium quality comment, and a low quality comment, and it gives different points to those different ones. And likewise with the responses to comments. And so if you leave a few high quality comments and you respond to a comment or two, that's usually all you need to get the full score. But then I've also, in addition to that, made it as another opportunity is after at the very top of the reading and the very bottom of the reading, there's a little next item tab that if you click on, then it will take you to 3 kind of comprehension questions that you can also score points on. So if you skip those, that's fine. But so a combination of reading and answering those can give you that full, that full score. And the full score is a full concept check. So that's perusal. So you can see how well you're doing in the perusal grade book, but then it takes a little bit of time to sync to the Canvas grade book. So you may not be able to see your score in Canvas, but inside the perusal itself, that has a grade book, and you can see, oh, it gave me a 20 out of 30 right now. I'll go back and add another couple of comments or something. So I won't tell you exactly the formula of how it does the weighting, but you can kind of infer that by seeing how it scores you. And I release those scores immediately as opposed to waiting for the due date. So that's what we're doing instead of Thursday. And so any questions about that? Thanks for submitting the muddiest points. Those are helpful to me to read through and to sort of see those responses too. And so keeping them concise is also helpful, so I can make sure I can knock them out quickly, so that if I do need to make any changes ahead of time, I can. All right, so last time we started introducing evolutionary metaheuristics, in other words, population-based ways to search over optimization problems. So I know I used the artificial neural network as a running example, and that's just a lot of times because the students that take this course are often from Sky or from SEMTI, and they that is an easy example for a lot of them to cling on to. But if that is not something that is in your background, then don't worry. We'll actually cover the ANN and its bio-inspired roots kind of from the ground up in a later unit. So what you really should know at this point is, there exist engineering problems for which there are multiple degrees of design freedom, which we call parameters. And so, you've got, if I've got a chemical process, I'd like to sort of say get the highest yield out of this chemical process. And I can adjust the temperature, the pressure, and the humidity inside the reactor, for example. And I don't know what combination of those three is going to give me the highest yield. Now, I can set those by actually setting them on the real reactor and then try a bunch of things. Or I might have a simulation of the reactor, which I can set those in and try a bunch of things. Or I might have a mathematical model of the reactor that is still kind of costly for me to execute a bunch of times, but it's sort of implemented in math rather than in sim, and I could try a bunch of things. And eventually I could iterate over all those things and find a good choice of temperature, humidity, and pressure so that I get the highest yield out of my chemical process. Well, these metaheuristics are ways in which we automate that process. So instead of doing a brute force search over every single possible combination of temperature, humidity, and pressure, then it intelligently tries one or many, and then from the results of that, then chooses new ones and new ones and prioritizes the search until hopefully it finds a good choice. So gradient-based methods like gradient descent or gradient ascent, depending on sort of what your perspective is, that people use in numerical optimization is one way to do this. But there are a bunch of optimization objectives that do not have gradients. And so that's why we have to come up with other clever ways to search through the space. And so we focus on nature-inspired ways. And so right now, we're focusing on evolutionary ways to do that, where you start with a population of candidates, try them all out, compare them to each other, and then from that comparison, choose the next set of candidates and iterate over that. So ultimately, instead of getting one solution out of this process, you get a population of solutions out of this process. And if you're really clever about how you build your metaheuristic, that population may have diversity that's useful to you. So it might just be you take the best solution out of the population, and that is the solution. Or if in the metaheuristics that we'll see coming down the line in about a week or two, then we'll actually see that the population itself may trade off among multiple functional objectives. So you might get one member of your population is really good at objective X, but not as great as Y. Another is really good at objective Y, but not as great as X. And so you converge on a portfolio where then you can ask your stakeholder, you told me X and Y are both important. I don't know which one is more important to you, and you didn't really quantify that. So here's a set of solutions, some that are better in X and less than Y, some that are better in Y and less than X. Which one do you think works best for you? And so that's when we get into multi-objective. That's where we're going. But we have to start somewhere, so we're doing single objective. So that's where we've been. Were there any questions about that? All right, so today then I want to set up the kind of fundamentals for the genetic algorithm, which is the first one of these optimization metaheuristics, the first more sophisticated nature-based optimization metaheuristic that we cover. So today I'm sort of going to give us kind of fundamentals of PopGen. And so this is applies to the genetic algorithm and other evolutionary algorithms, but the lingo that I'm gonna give here is going to be coming directly from the nature from the biology. So these are terms that we're using that we've borrowed from biology. So this is, the context here are living systems. And I'll say under evolution. And we'll have to define what evolution means here in a second as well. Okay, so we know that the living systems, at least we know on this planet, have DNA in them, right? So DNA encodes sort of the programming of life. But what does that mean? How do we get from DNA to me? So we say that that all of the DNA is built up or stored in these chromosomes, which are sort of strings of genes, where these things we can just say are locations along chromosomes. And I'm going to emphasize locations here. So in every cell in your body, there is a string of DNA. So the DNA itself. So I'd say the DNA has got these four nucleotides, which these things don't matter so much. But A, T, G, and C, and they get clustered into, I'll call them words of 3 letters long that encode amino acids, or um, special, I'll say, special delimiters like stop. And so basically that is how, that is how proteins, which, so your body is made up of metabolic pathways. The dynamics of life have to do with a bunch of chemicals being built, reacting with each other, producing products that lead to energy, say, being released, that are able to absorb energy, that are able to, when energy is being released, to generate changes in energy, like motion, like the motion of my hand, and so on and so forth. And so all of that is built up. So these amino acids, and this is kind of deeper than we need to go, build proteins. And basically proteins are the chemistry of life. I'll say of most of life. And that's how you program a living thing, is by coming up with strings of letters that are grouped into words, which are parsed into proteins, and then those proteins interact with each other like lines of code in order to create activity. So that's fine. But we are interested in quantifying how we vary. I mean, the way the proteins that are produced in my body are different than the ones in your body. I look different than you. I sound different than you. And where does that come from? And and what is the importance of these differences? And so these chromosomes represent degrees of freedom, where each gene we can think of as 1 degree of freedom. Or if you prefer, 1 parameter. And so you can think of chromosomes as a sort of vector of parameters, genes. So, like when we get to the genetic algorithm, we're going to create artificial chromosomes, and those will be artificial ways to encode the decision space of an optimization problem. And we will go over this again of Tuesday on next week. But just looking ahead, where this is going is a decision variable in an optimization problem is a gene. And the set of all decision variables together is a chromosome. And so the chromosome is just how you organize your decision variables into a compact data structure that you can easily reference and create and destroy and pass on, just like DNA. So genes and chromosomes. But the particular values of a gene are, which, like the parameter values, it's like the difference between a state variable and a state. The things that you actually store in the variable, so temperature is the variable, but 15 degrees Celsius is the value. Those different ways that things can vary are alleles. So we're saying here that genes have variants, and those variants are called alleles. I'll try to write that a little cleaner. And this comes basically from the Greek for other or variant, basically. And so that's basically saying what an allele is saying is there's a location on a chromosome that could have a string of letters on it. So each letter is not an allele. Strings of letters are the alleles themselves. And there's a location, double tapped. There is a location where we might expect to find blue, green, red. We might expect to find 50, 75, 36. We might expect to find A, T, 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 T. We might expect to find U or G, 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 C, T. We might be able to find all sorts of different strings of things at this particular location. And each one of the things that we might find is an allele. So you might say that if there's a gene for whether your ear is connected right here to your head or has a gap right there, then we would say that there's a gene for ear connectedness, and there's two alleles, connected and not. So those are the two values that go into that gene. And the collection of alleles in an individual is called their genotype. So if a chromosome is a vector of variables, then a genotype is a vector of values. So, I could basically put that here is that a chromosome is a vector of genes, and a genotype is a vector of alleles. And so these here are variables, and these here are values. All right, so that's the lingo that we use. So if I'm telling you, as we'll get to next week, and we'll go over this again next week when we make it more concrete with the algorithm, but in a particular GA, if we're talking about the design of your chromosome, we're talking about how you're going to encode the many different ways in which your design problem can vary. Are you going to put, is there one temperature? Is there multiple temperatures? And so on and so forth. Are you going to put this in position one, position two? Are you going to separate these things? We're going to see there might be reasons why it will be good to keep certain variables next to each other in a chromosome and certain times, split them up. So this is in a kind of design of your scheme. But then when you actually are running the algorithm, then you're going to say, I've got 50 individuals and they all vary and they're all a different candidate solution. So each one of them will have a different genotype, which means they'll have different values for those variables that you've put in at these different positions. So I guess I could also say you could say this are your positions along a chromosome. Now, I just want to say one thing that's going to be different is nature is tricky in that genes, alleles do not all have to be the same length in nature. So you can have an allele that is 100 base pairs long, but another allele at the same gene is 150 base pairs long. And that's because nature has come up with codes that say a gene is starting now and now a gene is ending. And so that allows nature to, because it creates these brackets, it can create these variations in length, which allow some people's genes to be longer than other people's genes. Or I guess the gene is always the same, but the actual alleles, are going to vary in length because they've got that stop and that start. When we create artificial chromosomes, it's going to be usually more convenient for us to just say, that we're always going to put an integer here. It's not like we're going to say this variable is, you could say that, you could do the same thing nature does and say my gene is itself a variable length array that has, some sort of stopping and starting or something that along this string that sometimes I interpret these for, like I don't actually know when my gene is going to stop. It seems a little silly because we have other ways to do that bookkeeping in a computer. We can actually say, well, this is just a pointer to some number or something like that. And, so I just have, so my chromosomes always, my genotypes are always the same length for all my individuals. But I just don't want you to get the impression that in nature, alleles always have to have like exactly the same amount of base pairs. It will seem like that as we build artificial chromosomes. We're like, wow, this is how nature does it. Then, what happens if there's a mutation that, splices in a few more, a few more nucleotides? Does that just screw up the whole gene? And it turns out no, because it actually doesn't go for a fixed number of nucleotides. It goes up until it gets a stop. So that's kind of a cool thing that nature does that we don't have to worry about. But at least when I started to learn this stuff, it was confusing to me to think about, well, how, does nature, like, how does nature decide that this gene is 100 base pairs and this gene's 150 base pairs? And how the heck did they ever differ? But it's because the lengths of the alleles don't have to match because of this stop and start mechanism. So any questions about that? Pretty clear so far? Okay. So, all right, so alleles, so populations uh, of the same species have distributions of alleles, not genes. We all theoretically have the same locations along our chromosomes, but we vary in the alleles that go in those slots. And so when we talk about evolution, we're talking about the change in the distribution of those alleles over time. That's all evolution means, the change in allelic distribution. So I'll just put that down here. So evolution is a change in the distribution of a population's alleles. And so what could cause those changes? Well, one of the things that could cause those changes is natural selection. So is the that some combinations of alleles provide more performance than others. And so those individuals that have that combination tend to have more offspring, and those offspring tend to have those alleles. And so then you get an increase in the distribution of those alleles rather than other alleles. But there are other things that can cause changes in alleles as well, which we'll kind of talk about here. Um, but so as an example, so like from parent to offspring, you can get mutations in genes which lead to different alleles. And this happens at a very low rate, typically. So that's one way that alleles can change, is that you just happen to have that, even though one parent organism had a particular allele, even if they cloned their offspring, there could just be a random change in a couple of letters, or there could be an error in how things were copied. And then you can get a change in that allele just by an introduction of that error. So that's sort of spontaneous, and that may lead to an offspring that is better performing than the parent or worse performing than the parent. But that change has nothing to do with the performance. And if you were to douse a population at a bunch of radiation, you might get a massive change in the alleles in the next generation of that population, which has nothing to do with selection. It's just because they've been baked in that mutation. Interestingly enough, there's a population of dogs that lives near Chernobyl, the famous nuclear reactor that melted down, and they do notice that those dogs show different traits, a term I haven't defined yet, and they've assumed that that was because they exposed a bunch of mutation. But it turns out that a recent paper that came out, I think last week or so, showed that the mutation rates, there are ways to measure this stuff in the dog cells, don't look any different than dogs far away from Chernobyl, which suggests that the traits that they see, the differences there, might actually be traits that were under selection. So they weren't just randomly generated by mutations, they were actually traits that for some reason in that area, the ecology is just different. And so those dogs that behaved differently actually were more fit. So it wasn't totally random. So mutation is a way of introducing novelty and randomness, but it can be very harmful at high rates because it's so random. So that's one way that the allelic distribution can change. But the other way, or an other way, is recombination. And this is particularly in when parents have sexual reproduction. So I'll skip over here and say recombination. And so that's when you've got a parent, two parents with two different genotypes, and they basically swap portions of their genotypes to produce offspring. And this is done internally in during it's actually during so this is done internally through a process of what's called crossover. And so these two terms in this class will often be very much associated with each other. And this is when two chromosomes link up, pick points to join at, and swap genes between points, otherwise known as a crossover. So you can imagine you've got this chromosome, you've got this chromosome, they pick a point, and then maybe I should have drawn those in different colors. So you could imagine I've got maybe my magenta chromosome here, maybe of this green chromosome here. There's this point, and then when they split apart again, then everything at the top becomes the genotype of an offspring, and everything at the bottom becomes the genotype of another offspring. And so you get a new offspring that's got a genotype that looks like this, and another offspring that has a genotype that looks like this. So these are parent, two parents, crossover, and then you have two offspring. And effectively, this interpolates between the two parents. So if the genes for, let's say, eye color are are over here, this is where eye color is encoded. And if the genes for height are over here, this is where height is encoded, then you might get an offspring that's got the eye color of the magenta parent, but the height of the green parent, and vice versa down here. And so that's what we mean by crossover. It is a particular operator that occurs during recombination, which is just a way of creating variation, not in quite as random and haphazard a way as mutation. So this is another way that if if you only have one offspring, so if this one gets discarded, then this can also lead to a reshuffling of the alleles, and so a change in the distribution of alleles. So that's another way you can get evolution, so a change in those things. But these are really, this is just, another way to generate novelty, another novelty generator. But really the thing that ramps up the changes in that ramps up the changes in alleles is natural selection. And in order for us to talk about natural selection, we have to talk about fitness. In order to talk about fitness, we have to talk about phenotypes. So this is just things that can happen at the gene level. But now we have to talk about, okay, great. Once we have the genes, what does that mean in the outside world, in the real world? And how does that then feed back onto the genes? So phenotypes and characters. Phenotypes, characters, and traits. I'll say and fitness. Okay, so the the genotype is just a code, but there is a function that basically maps. So when you take your genotype and you combine it with the environment, it produces what's called a phenotype. And these are the kind of functional consequences of gene expression. Or I'd say this whole process, I'd say, is called gene expression. And so the phenotype are sort of the observable consequences. And we'll make that more precise here in a second. But I mean, basically, the thing is like, you might have a gene for, let's say, blue eyes. But let's say that in order for your species to produce blue eyes, it needs to go out and find a pigment in nature that exists in a particular blue flower. And it has to eat that flower to get that blue pigment. And once it has that blue pigment, then that gene can now build blue eyes. So if you can't find that blue pigment in the environment, then it doesn't matter that you've got the gene for blue eyes. You're going to get some other colored eyes. So it's not enough to have the gene for blue eyes. You have to have whatever the environmental precursors that need to happen to also get that to happen. So these are sources of variation here, is that you can get variable expression due to variation in your genes, but also due to variation in the environment that actually is part of the machine, which generates those expressed genes. So you're not going to be perfectly, your height is in part, a function of your parents' height, but it's also in part due to developmental consequences of like, did you grow up in space? Did you grow up in a high gravity environment? Did you eat enough calcium to create, certain bone growth and so on and so forth? That's why they'll tell, like, oh, this particular thing, if you feed kids, will stunt their growth. Well, so that is an admission that environment matters for phenotype. So, so that environment could also include development. I'll just put that in there. So it's not just the stuff that is immediately available, but it's also your developmental history. So gene and environment come together to produce phenotype. So you can't see someone's genes. You see the expression of those genes. But someone might be tall because of environmental factors, or they might be tall because of genes. So just because you see that they're tall or they have blue eyes or whatever doesn't mean that you actually know what genes they have. So there is not a one-to-one mapping between genotype and phenotype. So, and I'll say, I'm go write this, produces. And so so basically what your phenotype is it is a set of traits that you show, but so yeah, okay, I'll say that your phenotype is a collection of trait values that are observable and produced in part by genes and in part by the environment. Okay, um, so I can, like, for example, a trait might be blue eyes, or trait value. I would say that a character might be blue eyes, and this is for the trait eye color. And so this here is a trait value for eye color. So there is this like weird, it seems it's very closely related to genes and alleles and so on. We can define kind of slots of variation, observable variation. How does a human, what are different ways that humans can vary from each other? Well, in eye color, they can vary in eye color. So that's one dimension. But what are all of the different eye colors that are available? Well, those are the particular, this is like, well, it could be blue eyes and brown eyes and so on. So a lot of times these terms will just be used kind of interchangeably, and that's fine. But what you have to keep in mind is there is no one-to-one mapping between particular alleles and particular trait values. So that is something that you can, it's easy to say like I have the gene or the allele for blue eyes, but that allele might actually be also involved in blood pressure. It might also be involved in height. It might be that it's you, if you have that gene and the right diet, you get blue eyes. But there's all sorts of things that go into the actual production of these things. So we can't just say there's this one-to-one mapping between like this observable thing is encoded by this and only this. This observable thing is encoded by this and only this, and so on. There is not that mapping. There's just a set of code, and then there's a set of functional observables. And the code produces the functional observables, but in a non-trivial way. Rarely do you get an easy mapping where you can say, oh yeah, that thing right there, your hair color, whatever, that is exactly coded by this thing here. So if we hack that out and change it to something else, then we'll produce different hair color and nothing else. Rarely is that the case. So there is not that single genotype to phenotype mapping. So in reality, there is there's a complicated function which maybe I'll call lowercase p for it sort of arbitrarily, that takes your genotype and it takes your environment and it produces your phenotype. And this is shortened by G by E to P, what people will say. And so there is no way to say that like, oh, this gene or allele corresponds to this trait. This does not, that is not, this is not often do you get this case here. So it's this more complicated mapping here where some genes are involved in multiple traits, and certainly the environment is involved in the expression and so on and so forth. And it will be the same thing in really complicated problems. Like if you've got lots and lots of decision variables, it may be the case, and certainly with neural networks and things like that, it's a great example of it, that particular genes, so particular decision variables, may actually have effects on multiple aspects of the function that you're measuring across those individuals. And so you don't just want to get into the trap in thinking that, oh, I need more of this. Well, I just need to then crank up this decision variable. Your algorithm is actually going to be searching. It's almost like the genotype search space is totally different than the phenotype search space. And they are clearly related, but it is not a simple just mapping of whatever's happening here is exactly what's happening there. So, yeah, question? Mm-hmm. Well, there are actually very few genes where there's absolutely no variation that comes from the environment. So it like the blue eye thing, if you can't express if you have if the physiology that produce blue eyes requires a pigment, then even if you've got the gene, like you're the only one that can produce blue eyes. But if you still don't find the pigment, you won't. So there could be other preconditions and other variations in that. So you find that a lot of birds, for example, have really bright coloration, which is encoded in their genes, but they can't display that bright coloration unless they find these particular proteins in nature. So when they show really bright colors, they're actually signaling to potential mates that they've been able to go and find those things. So it's actually a conspicuous signal that they have high quality because not only do they have the genes, but they have not only the genes to display the colors, but they have other genes that allowed them to survive to find these difficult places to get these pigments that then now they can wear like a coat of arms around them or so. Absolutely. Um, Human suntan is an example of this as well. So, you can have significant variation in your skin color over, weeks. And that just comes from how exposed you are to the sun. And then likewise, if there's if you start eating certain things that actually might, there's certain drugs, for example, that can, as a side effect in small amounts of the population, turn the skin blue. And so there are definitely ways in which traits that you view as static actually have that aren't fixed throughout their whole life. Now there are traits that are fixed throughout your whole life, but the developmental environment when they first were fixed still matters. And so there might be some other trait that that is not 100% encoded by a gene. It's a gene plus something that you were fed exactly when you were 1 month old. And if you didn't get that thing when you're 1 month old, it sets up a sequence of dominoes to fall that for the rest of your life, you're stuck. So yeah, it's a big part of what you have to do in biology is try to tease out the variation in this phenotype. So, this I'll call this delta phenotype, the variation in phenotype is, there's always the question of does it come primarily from the variation in genotype or variation in environment? And so there's all of these experiments that they do, cross-fostering experiments and things like that. We're going to take a bird from this area and we're going to get an egg from this area and we're going to put them in the nest of a bird of a different species in this area. And then we're going to see how much of the adult bird looks like their sister that stayed in this area. And so in that particular case, you could see that, well, if they look identical, even though I've completely changed the environment, it's got to be all genes. But if I've now gone over here and this bird that looked like this tiny little thing that I could hold in my hand is now this Tyrannosaurus Rex, then we're like, okay, well, then probably environment. And so, but of course they interact because the environment probably turned on genes, and so on and so forth. And so this is very complicated, this mapping. So we just can't kind of assume that you never really can say you have the gene for X. You're like, well, you have a gene that in the right environments produces X. Yeah. Mhm. Yeah, in fact, we would bin that into environment. So this is supposed to say environment down here. So really, environment or biology is about explaining variation. And so you observe, so you're going to observe a certain amount of variance um, in measurements. And so, so you got var, phenotype. And then so the question is, there's going to be some scalar of var genotype and plus var environment. And part of what you do is try to find these coefficients. And so you might be able to totally control the genotype, but if once you've controlled for the genotype, so you've created lines of mice that are all completely identical, so there's absolutely no variation here in the population you've created, so this drops to 0, then everything that's left over, we bin into environment. And so environment could be the outside environment, or it could be some sort of weird accumulation, like it might be that when you got born on your head, and I got born on my feet. And so that caused, during some important developmental process, all the cells pulled into your head and pulled into my feet, and that totally changed our development. So there's kind of like an internal variance, which is like a memory of some external, change in initial conditions. So so then the next question is like, well, can you then split up these environment into different, this is variation in development, this is variation in something like, if you can bin all these up, then you can have different coefficients on each one of these. And then you gradually try to control for each one of these to assign the right coefficients. And that's largely all biological experiments are setting up exactly this to some extent, is control for some variance, see what's left over, and then try to account for it, try to split it up. Does that answer your question, or is that convincing enough? That's at least where we put the bookkeeping. We call that an environment. But random chance, whatever the mechanism is, would be into one of these sub bins. Yeah, there's always residual. Yeah, okay, so that's so all that's fine. That's how you get to phenotype, but then we still haven't got quite to fitness yet. So once you're starting to express things, then there's a possibility for the thing that you've expressed, the phenotype, the traits that you're showing, to have an impact on how many offspring you're allowed to have. And those offspring will then have a much greater chance of having the alleles that you have than other sorts of alleles. And so that can change the distribution of the alleles. And so there's sort of an additional function here. We'll call it, this is like a fitness function. And so it is a function of phenotype, where, of course, phenotype is a function of genotype and environment. And the fitness is basically equal to or proportional to, so prop to, the number of offspring an individual has. And we often refer to this as on average. So we're saying that an individual, because of the variation that comes in here, then we're saying an individual with this genotype produces this many offspring on average over their lifetime. And that's what we refer to as fitness. It's a relatively, it's not meant to be a pun, but it's a relatively simple definition, because really the interesting stuff comes in how then fitness compares. So if every individual has the same number of offspring, they all have the same number of fitness. And so there's going to be an equal chance that anyone's alleles will get into the next generation. But if there's some traits that tend to make some individuals reproduce more successfully, then they will produce more offspring relative to everyone else. And in the random draw that is life, then they will have a greater representation in the lottery that will be the next generation. And that is natural selection. So basically, we've got, we start with genotype. And it produces a phenotype, which produces a fitness, number of offspring, which might go through some sort of limitation. And so, there might only be enough food in the environment for 100 individuals. And you started with 100 individuals, but everybody had the opportunity to have offspring. So now there's 250 individuals. Well, only 100 of them are going to survive the food limitation. So which 100 survive? That's sort of an example there. Or it might have happened, in the actual production of the offspring is that in order the additional offspring that were won by a certain phenotype ended up costing offspring of another one. So there might be limited mating opportunities. So the limitation could also be built in, here. But the point is somewhere in this pathway, so somewhere here, then there is a limitation that is going to make it beneficial to have more offspring than less offspring. And so that is going to then feed back on the distribution of alleles on basically genotypes. So this will change the distribution of alleles. And that right there is evolution by natural selection. Or that's the kind of new term here. So there are a bunch of different ways that you can have evolution, but this particular process is one of the ways that explains changes in alleles. And it's because that certain alleles provide more fitness, which means there's a greater chance for those alleles to get passed into offspring than other alleles. And thus you get this thing moving around. So it's not a perfect process. So due to things like the way the chromosome is set up, then you can have alleles that, due to which genes they sit next to, if it is very unlikely that sex will break those two things up, then if you've got one allele that provides a lot of benefit, it might just have on its coattails some genes that are next door that if they just happen to have a greater association with that allele than other alleles, then they might too see a change in their distribution, for example. So the genomic architecture really does constrain how well this process can shape things for the best. So, and that's another, complication in population genetics. But generally speaking, this can cause that at least certain really strong alleles, certain alleles that have huge fitness effects, can lead to a change, an increase in those alleles and a decrease in the kind of alternative alleles. And that's what we mean by natural selection. So questions about that? All right, so then I just want to bring up one last process that of evolution that we have to worry about a lot when we think about GAs, and that is the process of drift. So, right, let's try this guy. There we go. So imagine if you had a population, and I'm just going to keep it simple, and there were two alleles in the population, A and B. So two alleles, and they're equal fitness. So there is no benefit to being A and there's no benefit to being B. Well, if that's the case, and let's say they have equal fitness, and let's say for both of them, f of x is equal to, 1. So in that case, then in the next generation, they get to stick around and they produce an offspring that looks just like them. And so here, you still have the same distribution of alleles, just more individuals. But if you do have a a limitation in the environment, so let's say only enough food for 4, then there's no fitness difference between these. So that means that this population is going to be a random draw from these individuals. It is unlikely that this random draw is going to have an equal distribution anymore. So you might now get A, B. And again, these are equal fitness. And so this same process might happen again. So you could get reproduction here. So now you've got AA, BB. But now, if there's only enough for 4, so we got our limitation to 4, then you can see it's very, very likely that the next population will have all A's. And so this is a natural state. This is a stable state of this random process. So it's an absorbing state of what you can model this as a Markov chain, and the all A and all B states are two absorbing states of this Markov chain. So this will probably become all A's. And if it isn't all A's immediately, it's very likely to eventually become all A's. It is very unlikely that B is going to start randomly growing and in its proportions again and eventually become all B's. And so this is what we refer to as genetic drift. And this happens when there are very little fitness differences. And it's particularly strong for small populations. And the last term that I want to introduce here is the term fixation. And so fixation is this absorbing state. When a population has purged all of its genetic variation, then we refer to that as fixation. And so genetic drift naturally ends in the population going to fixation. So fixation is when we have population with no genetic variation, e.g., all A allele. And this is sort of a natural outcome of genetic drift. And this is a real pain in the butt because right now A and B have no fitness difference. But your optimization objective might change a little bit in the future. And there might be something special about B where it will make it better adapted for those changes in the future. But now you've purged B from the population, and you're going to have to count on mutation bringing B back. And that may be nearly impossible. So we're going to have to figure out when we build our artificial genetic algorithms how to balance between selective pressure and drift, because the more selective pressure has the effect of purging fitness differences. Because if you've got a really bad performing genotype, it is likely to get taken out. So eventually you get a bunch of genotypes that are similar in performance. And so that's exactly where drift is. So you get selection purges differences in fitness, but once all those fitnesses differences come over, drift comes in and purges variation that is left over in the genotype. So you all get one genotype, and you've lost out on whatever innovation was latent and sitting in there and just waiting for the next optimization objective. So that's what we'll pick up next week as we build the new genetic algorithm. Like I said, Thursday, class is canceled, but we do have that assignment online instead. So take a look at that, and I'll see you Tuesday.